Hi, I'm Zoe.
I'm Alice.
I'm Don. Today we want to tell you about a tumor suppressor called p53.
p53 is a protein that regulates cell division and therefore is a player that helps an organism avoid cancer. They're encoded in our DNA and are synthesized through an intermediate mRNA. So what is a protein? Proteins are molecules that work together to help the cell divide and survive. Our cells go through cell division to proliferate and pass on genetic information. These regenerations allow development of a single cell to a child and eventually an adult. However, once in a while, our cells make a mistake or they are attacked. The result is damaged DNA. This damage leads to accumulations of mutation that can cause the cell to proliferate out of control and lead to cancer. Because of this, organism has developed a response to protect the cell. p53 is the most significant protein used. It can halt cell divisions or even lead to programmed cell death. An error in the functions of p53 is the leading cause of cancer. In fact, this accounts for more than 50% of human cancer.
Broadly, the way in which p53 is initiated is due to the removal of certain proteins attached to p53 that keep it in the cytoplasm and away from the nucleus where DNA is located. Without that protein, p53 is able to move into the nucleus until the DNA to create specific proteins that stop the cell from dividing. p53 is heavily regulated in multiple ways, such as ubiquitination, in which p53 is broken apart, phosphorylation, in which MDM2 separates from p53, and acetylation, in which specific genes are told to create their proteins. Here, we'll go a bit more in detail over each of these mechanisms. First, ubiquitination. Attachment of MDM2 to p53 keeps them from the nucleus and, if joined long enough, adds a ubiquitin chain. These ubiquitin chains signal to break p53 apart. To return its function, MDM2 must be removed. This is done by phosphorylation, or the addition of phosphate groups by a kinase protein. ATM responds to DNA damage by activating CHK2 and deactivating MDM2, both through phosphorylation. p53 is able to make copies of itself when it is phosphorylated by the active CHK2. Finally, there is acetylation, in which p300 attaches an acetyl group to p53 in the nucleus, as well as to the DNA surrounding the genes whose information is needed for halting the cell cycle or causing cell death.
Clearly, p53 is important for protecting the cell against DNA damage. But what happens when p53 stops working? These mutations can be very harmful to the cell, leading to uncontrolled cellular proliferation, or in other words, cancer. But what about when p53 doesn't stop working? Without MDM2, p53 will bind to everything and cause a halt to the cell cycle or cell death. This is extremely harmful during development when cells must divide constantly. As you can see, p53 must be kept at a high standard of regulation because incorrect regulation in either direction can create harmful results that in both cases have the potential to lead to death of the organism.
Thanks for watching.
We hope you are able to gain a better insight into what p53 is and why it is so important into the regulation of the cell cycle.
Bye!